Hi everyone, I'm Farzaneh Najafi. We have another session of Sybeco today. We're hosting Dr. Jennifer Bussell. Jennifer is an associate research scientist in the laboratory of Dr. Richard Axel at Columbia University's Zuckerman Institute for Mind, Brain, and Behavior. She received her undergraduate degree from the University of Chicago in biological sciences and her PhD from Rockefeller University with Leslie Bussell. She studied the neural circuitry underlying information seeking and curiosity and is interested in how the value of knowledge guides behavior. Thank you so much, Jennifer, for accepting our invitation. We're so excited to have you.
All right. Well, thank you, Farzana, for this awesome opportunity. This seems like a really cool lecture series. Everybody can hear me okay? All right. Okay, great. So, as she said, I study the neural basis of the desire for knowledge, and I'm trying to tease apart the neural circuit that underlies seeking information based on its intrinsic value rather than extrinsic reward. And so humans and animals seek information. We're constantly taking action to gain knowledge about the world and improve our decisions to maximize reward. I like to start with this gorgeous painting from Gerhard Richter. And this woman is totally engrossed in reading her magazine. And I like this because it illustrates that acquiring information helps us make decisions and get rewards, but it also can itself be pleasurable and rewarding. And so we seek information and want knowledge in the world, not only because it leads us to external rewards, but also we have this innate desire to get information that can be intrinsically valuable. And this is also true of animals. So these early experiments from Tolman and colleagues, I think they're illustrative. And we've known for a long time that animals acquire information in the absence of external reward. And so the way these experiments worked is that there's a maze and a rat is placed in the maze and it explores, it runs around the maze in the absence of any reward. Then when the reward is introduced, the rats that had an earlier opportunity to explore go right to the reward because they have gained knowledge of the maze and formed a cognitive map through this latent learning process. And so I'm pointing to this because it illustrates two things. The first is that information has intrinsic value to a rat. It acquires information in a context completely without external reward. And this is innate. The rat doesn't have to be taught to do this, which suggests that this is this evolved innate drive. And two, this suggests how such a drive might have evolved because the information does end up being useful later. And so this suggests that our desire for knowledge maybe evolved such that the brain, almost as a heuristic, assigns intrinsic value to information to incentivize information-seeking behaviors, because usually with a long enough time horizon, information is useful. It certainly has been useful in sort of the evolutionary past, and this now generalizes such that we have this tendency to seek information. And so even since Tolman, many other people have looked at this tendency to seek information, particularly in this context of it being non-instrumental and disconnected from gaining additional external reward. And so many different people have looked at this in both functional imaging in humans and in electrophysiology in monkeys. The brain's generalized reward system has been shown to respond to information as if it itself is a reward. And so this is an example, oops, I'm pointing with my mouse on my screen, an example, midbrain presumptive dopaminergic neuron in a monkey. And the same neuron is responding with a prediction error-like signal to physiological juice reward and to the prediction and receipt of information. And so these kinds of data imply that the same reward system, and here, in fact, the exact same neurons that reinforce actions to gain physical juice reward could reinforce actions to gain information. And this poses a really interesting, difficult problem, right? This idea of information as its own reward. And that is, how does the brain know what information is? How does it identify and assign different values to potential sources of information like these books? So external or extrinsic rewards fulfill physiological needs, and they elicit reinforcement through physiological processes that activate specific ensembles of neurons to elicit reinforcement. So, your tongue and your gut physically know that you've eaten an apple and convey its value to specific neurons in the brain to elicit reinforcement. But information is different. It's defined by cognition itself. If we take as kind of a rough and ready definition for information from Shannon, that information is that which reduces uncertainty. In that case, like our desire for information, which things are most informative, depend on current knowledge. And so this process of the brain recognizing information and assigning it value is kind of an elusive process of metacognition. And moreover, if it's the same reward system and even the same dopaminergic neurons that can respond to information as a cognitive reward and physiological rewards like juice, that suggests that there may be other unique pathways that recognize and assign value to information. And so that's what I was interested in studying. That's what this paper and this project is about, is how is this kind of unique value of information computed and represented in the brain? And we started to dig into this by asking if we could discern and see a representation of intrinsic information value in the mouse so that we can then use the tools of rodent systems neuroscience to kind of trace and crack into the circuit at a mechanistic level. And so the paper and what I'll tell you about today starts with first asking whether mice seek information for its own sake. Then if there is a representation of that value of information in the mouse cortex, if that representation and information value is in fact distinct from the representation of extrinsic physiological reward value. And then there's this last, maybe more elusive and exciting question that I won't really answer today, but future work is kind of aimed towards, which is how might such a representation arise? And so there'll be two parts of the talk, and there's really two parts of the paper, which is kind of the behavioral paradigm we developed to ask whether mice seek information, and then the imaging work that we've done to look for representations of information value in the mouse orbitofrontal cortex. And
Question,
obviously, yeah.
So when you say seek information, is that similar to saying seeking novelty? Like, can we have a definition of information a little bit here?
Yeah, so this, I mean, kind of within the field, this becomes a little bit tangled, and that's why I kind of posed the rough and ready definition as information is that which reduces uncertainty, because then when you talk about kind of desire for information or curiosity in almost a lay term, that ranges a lot and is a little bit hard to define. It's different from novelty, certainly in our task, because information is tied to a specific probabilistic event. And so, like in the task I'm going to talk about, information just reveals a future outcome. And so it's not novel in the sense of having like never been experienced before, but it provides a novel cue about the future on each trial, right? And so novelty factors in a lot in terms of, often people think about it as like one of the things that alerts you that something might be informative, right? But not everything. There's, I'm gonna get sidetracked here, but there's a classic problem of the television static, right? In that it's constantly changing and giving you like novel images, but it's not necessarily giving you any information, right?
Right. So I guess the key definition here is really reducing uncertainty. So, and that's how we can disentangle that from novelty.
Yeah, I think so. And you'll see, I mean, in the task that we came up with, the mice are incredibly overtrained, right? Like there's literally nothing novel about what's happening. It's just that they don't know whether or not they're going to get water on that trial, and the information tells them that.
Right. Because I think novelty even increases uncertainty. It's like, yeah.
Yeah, but there's this interesting paradox with novelty where like too much of it is bad, right? So totally unfamiliar things are often frightening. There's like neophobia and fear of novelty. Totally familiar things are often boring. And there's kind of a U-shaped curve where in the middle is like optimal in terms of kind of triggering like interest and curiosity.
Interesting.
Yeah.
Thanks.
Yeah. Okay. So we took, and like this has been studied in both humans and non-human primates, and even there's early kind of behaviorist experiments in like pigeons and rats using a paradigm for non-instrumental information seeking where information resolves an upcoming probabilistic uncertain outcome. And we've modified that framework for the mouse with odor cues. And so the mice are water deprived. On each trial, they enter a center nose port. And in that port, they receive an odor on each trial. One odor directs them to the left reward port, one directs them to the right reward port. For a particular animal, say the left reward port is going to be the information port, and the right port is the no information port. And these side reward ports, both of them deliver water reward with an identical 25% probability. When the mouse enters the side port on the information side, it receives either odor A, which is always followed by water reward and signals a rewarded trial, or odor B, which signals an unrewarded trial. On the no information side, the mouse also receives an odor when it goes there, odor C on 25% of trials, odor D on 75% of trials. But these odors at the no information side give the mouse no information and about the eventual water reward. They're each equally followed by 25% water reward probability. So the two sides have identical water value, water reward, but on the information side, the odor tells the mouse if it's going to get reward or not. And once the mice have learned this task, after training for several weeks, we introduce a third free choice odor on a third of the trials. And in which case the mice can get water by going to either side. And we're able to measure their preference and their seeking of information by what they do on those trials. And so with any, yeah.
Just making sure just the task. The part about information, no information, I guess, is clear because in the information case, one odor is 100% linked to reward, and the other is not. In the other case, both odors are 25% linked. The initial choice of left versus right, I missed that part. Was that also odor driven?
Yes. So their initial choice, they get this kind of initial odor cue in the center port. And there's three trial types. There's like forced or directed information. You can only get water by going to the information side. There's forced no information. You can only get water by going to the no information side, or there's a free choice odor. And we get to the neural data, like it was the responses to these odors that kind of differentially predict information that we got really interested in.
So tell me if I should wait and if I'm being rushing.
No. If everybody doesn't get the task, we've got nothing to talk about. So
thanks. So the force there are three different odors presented in the center port.
Yep, one on each trial.
Right, but what do you mean by so it's at the end of the day, the mouse's choice whether they want to go to left or right. So I guess.
On the choice trials, they can go to the left or the right to get water. But we call them forced trials because when the mouse gets this odor cue, it can only get water by going to the information side. And so we kind of train them up and teach them that odor means you have to go to the left.
Right.
And what happens if they don't go to the left? Like, what if they go to the right anyway?
Then they have to wait out the duration of the trial because that's like an incorrect trial.
So if they go to the right on a forced left trial, then there is no further odor?
Correct.
Okay.
Yeah, and then they have to repeat that trial, right? Because the idea is, and we want to be really careful, and we kind of keep track during their training that they have equalized rates of water reward like per time and per experience of these center odors so that they can't be choosing or differentiating them by like, oh, I've gotten water a ton by going over there. I like it more, right?
Awesome. So to reiterate, force information, the right side will do nothing, and it will be punishment, basically, like timeout.
Yeah, timeout.
Force, no information, the left side will do nothing.
Correct.
And free choice is when either right or left, they will receive one of these two, information or no. Okay.
Yep.
Awesome.
And then we put another wrinkle in where within each animal, after kind of training them and measuring preference for a couple of days, we reverse the sides.
Wait, sorry, Jen, before the wrinkle.
Yeah.
Can you go back? So why is it 25-75 for the rewarded odor and the non-rewarded odor instead of 50-50?
We wanted to, it turned out to not matter that much, but we wanted to control for, because we're going to look at the neural responses to all these odors, we wanted to control for the fact that this odor is less frequent. And so we made the kind of frequency of the presentation of these control odors match. And the reason we did 25% reward and 75 instead of 50-50 is actually like max uncertainty, right? We did 25-75 because there was old literature in rats that information about rare high value rewards was more valuable. And it gets sort of hand wavy about like why that might be, but because in designing this, it takes so long to test everything, and we really wanted to maximize the chances of eliciting this kind of behavior in the mice, we went with that.
I guess it also gives you more trials because they have to do more to get the same amount of water.
Yeah, that's true.
Okay, got it, I think. Ready for the wrinkle.
Okay.
But then to follow up on that, but then they are more excited about the left side because they have higher chance of getting reward as long as they recognize odor A.
Yeah, so once they get odor A, they are for sure getting reward and they're happy and everything's awesome. But at the time at which they're making their choice, the reward probability is the same.
But the reward probability, so if it is, so the force choice so those are not cued, right? Would the mouse know whether this is a force information?
Yes.
They're cued.
Yeah. So they've learned, like the way we train them up is they've learned that this odor means I only get water by going to the left. I have to follow that odor.
Sorry, it was by odor. Sorry, I forgot that an odor basically cues it.
Yeah.
Right.
Yeah.
But then
so
as soon as they receive that left odor, which is the information odor, they know that they're gonna get reward.
Yeah.
But
it's exactly that. And they're happy, and that's awesome. That actually happens. It's more frequent, much more frequent that they get this odor that means, oh no, I have to wait around for another, and this delay is really long. I should say it's 10 seconds, right? So it's, oh, I have to wait forever. I will not get water on this trial.
But I feel like what Farzaneh said is not matching my understanding. Farzaneh said when they get the left odor, they are more likely to get the reward than the right odor, but I don't think that's
not correct. Exactly. When they get those odors, the reward probability is the same. It's when they get this odor on the side that predicts water that, yes, at that point, they are definitely getting reward on that trial. That only happens on 25% of the trials on the left side.
So are you saying that odor A is presented not 50% and B 50%, A is 25% and B is 75%?
Yes.
Oh, I'm sorry. And your arrows actually do show that, but then your arrows C and D are also thin and thick. Should those then not be equal?
No, so C is actually also 20, and that was the question Chris asked, right? So C is 25% and D is 75% also. It's because we wanted to control for the fact that A and B are at different
Okay, now I understand. I thought that Chris was
Yeah,
asking about that. Yeah, it took me a bit longer, but I guess now I understand, right?
Yeah.
Okay.
Okay.
But then if you do the math here, the 25% with that 25 plus 75 times that 20, so that sums up to the same value?
Yep.
Okay.
Yeah, because I actually, even in like the code in the task, right? Like the water reward on this no info side is totally independent of which odor they get, just at 25%.
Awesome. Task is designed well, not as
There are things that I like about it and things that I don't. Okay, but then the other wrinkle is that within each animal, we reverse the side locations so that the mouse that has been, following and going to the left to get information, now if it really wants the information, this is uncued, right? They have to start on the choice trials going to the right to get the information. That's to control for side bias. Mice are sort of notoriously repetitive and side bias, right? Even though these trials are like interleaved on a third of the time. Okay, so what do they do, right? Oh, and again, there's a 10-second, it's quite a long delay in between the side odor provision and the actual water reward outcome. And we tried to control really carefully that they can't avoid trials. They cannot make things go faster through their actions. Everything is time locked such that once they've indicated their choice, things are going to happen at the same rate and the water reward will come at the same rate across the two sides.
And
How did you get them Oh, I'm sorry. I just always have questions about the mouse behavior. How did you get them to wait 10 seconds for their reward? Like, how do they even know that they have to wait 10 seconds and then a reward comes out?
Yeah, so we shape it up, right? So the delays start low, and then and there's an earlier training stage, which if we have time, we'll get to in the end, where the way we shape the task is that first they're just following odors to the left and right and getting reward immediately, and then we slowly increase that delay. Once they know that, oh, I have to wait around a really long time for 10 seconds, then we make the rewards probabilistic, introduce the information, then we add the choice odor. So it's a long, it's a
Very long.
weeks-long process.
And after how long of training on one contingency you reverse it?
In some of the animals, we tested preference in kind of the final version of the task for 3 days and then reversed for 3 days. In the imaging animals, because we wanted everything to kind of like stabilize and be quite set, we did like 6 days and then 6 days.
And how long does it take total for mice to learn this?
Several weeks, right? Like I think the best animals can do it after like 3-4 weeks, and then some others take a bit longer. One of the things in the training is that we don't have a lot of super great readouts that they understand the abstraction of information prediction. So we kind of overtrain that stage and have them kind of just doing information, no information trials for like a week just to make sure that they understand.
And these are black 6 mice.
Yeah, males and females. We didn't see
we
any
use
differences.
auditory and visual stimuli, and I feel like with those stimuli, this would have taken forever. So I think the only reason your mice are learning this is that it's olfactory.
Yeah, no, I think there's a number of advantages to doing it. I think freely moving makes it easier. It's more intuitive for them. The odor cues help. The odor cues are helpful both, I think, for learning the task, and when we get to the neural data, it was really nice for us that we know so much about the odor representations. Okay, so what do they do? They like the information. So these are the kind of 36 animals that we tested. There's a point with error bars for each individual animal. Here's the mean. Most of the animals had a reasonably strong information preference. These are data from the last 2 sessions on either side, balanced across the left, right. And we did a number of controls. They're kind of in the supplement. I'll skip them for now. But these animals that didn't really have info preference, we convinced ourselves that they were side biased. So they would, just pick the right side on the choice trials and continue going there across the reversal. But we wanted to make sure that the reason they're choosing this side that we've said, is giving them information, is that they want the information and not something else. And one of the things we had to consider is what they do on these trials when they are told you will definitely not get reward, right? Because they're getting the odor cue in this reward port, they're eventually getting the water there. On these trials, they know they won't be getting water, and they leave the port. So they're not required to stay poked in the port during the delay. What they do is on these trials, which are the majority of the trials on one side, is they leave. And we considered that mice might value that, right? Like even though they're able to leave on every trial, they might really value this situation where they're not staying poked in the port. And so we did another version of the task where we cued them on every single trial 2 seconds before the reward outcome, what was going to happen. Yeah.
Sorry, I thought I'm doing too many interruptions, but then I didn't want to go to the next point. So next plot, if you could go to the next slide. So the y-axis here, that is computed from the free choice trials.
Yes.
Am I getting, so you see on those how many times they go to left versus right. That's how this is happening.
Yeah.
Right.
And
Okay,
it's across the reversal. So it's how many times this one isn't, but
Right.
the previous one was. So it's, yeah, what fraction of those free choice trials are they going to get information?
Awesome. Yeah, that's it.
Yeah, and so in this version where our idea was we tried many, many different things to equalize and control for this behavior. What ended up working the best is if we cue them on every trial right before the reward happens, that way, if they want to, they can leave the port on every trial and then come back and get reward. They did that to some extent, and in that case, they still had a strong preference for the information. And so this convinced us that this is really about them wanting to know what's going to happen, not some ability to like go poke around in the corner. And so this choice preference suggests that information has value to a mouse. And we wanted to know if that at some level was the same kind of value as a canonical extrinsic reward reinforcer like water reward, and to do the kind of classic economic decision-making thing of will they pay to get information? And so we did these experiments of making the amount of water on the information side diminished in comparison to the no information side. And so here is the preference that the animals had when the two sides gave them equal water rewards. And then in kind of blocks of sessions, we decreased the amount of water they could get by going to the information side and saw that they still had a strong preference for information. Even when the information side is giving them only like 1 drop of water compared to 4 on the no information side, they're still going there half the time. And that suggests that mice are willing to pay to get this kind of information by giving up a huge chunk of their water reward just to know what's going to happen.
So that's a really cool result, Jen. I just want to make sure I understand it.
Yeah.
And by the way, cool to see this all coming together because I know you worked on this task for a long time, and it looks really nice. So I'm not sure if those water droplet icons are meant to be interpreted literally or not. So
they are. They're each like a 4 microliter drop of water.
So the break-even point for the mice, they would rather have information than have 4 times as much reward.
Yeah, it's somewhere between, depending on the mouse, it's somewhere between like 25 to 33% of the water value is that kind of break even, pay for info point. Coincidentally, that's like almost the exact same percentage-wise amount that they found for monkeys doing a similar task.
I would have felt like that's a very high differential, right? For information that's like not actually actionable in any way. Like I would have liked to think that I would be, I guess, closer to 0 on this scale.
Yeah. I mean, and it's surprising to us. And we did do, I'm trying to think, it varies to some extent across animals. It may be that you're hitting some kind of ceiling effect. And like if we did it long enough, right? Like these aren't sort of infinite length experiments. We kind of just went long enough to see that things were changing. But we do know that like in the case where the mouse is just choosing between 1 and 4 drops, they're never going to go to the 1 drop, right?
Can I add a point here? So if you see our hands, I wouldn't interrupt. I don't know so in our hands, when we give a large drop of water to mice, it may drop from the water spout. So did you guys control for that it's not about that they have actually full access to the full amount, and that's not,
Yeah, so it's like in the cases where we're giving 4 drops, it's a drop of 4 microliters and then like 50 millisecond wait and then a drop. So they are meant to kind of have time
to have it.
To get it.
So basically they prefer to know that I'm gonna get the reward than how much reward I'm gonna get.
Maybe. Or certainly there's some level at which they're willing to give up a decent amount of water to get this information. And either one can think about it to be in the delightful state of knowing what's happening, or to avoid the horribly aversive state of being uncertain about what's going to happen, right? It's kind of two sides to the same coin.
But I would guess that humans will, or some humans will behave very differently here. So I don't know if you had inter-mice variability, but I would guess some humans will really value also be more like greedy. I want to get more reward, even if it is less likely for me to get it.
Yes, humans, there's variation across our mice, there's variations across people. Things get really, really variable if information is about negative events. So here everything is reward or nothing. If you give people like a lottery task and it's you start out with some amount of money and you can either win or lose, there's huge variation in terms of if people are willing to choose information about losing, or how much, if anything, they will pay. And I have a version of the task that's not in this paper, but we've been working on a version of the task with mice where the information is about shock, or like a mild shock or no shock.
Mm-hmm.
And it's much more variable across mice in that case.
But here, basically, what this is saying is that mice, I don't know what percentage, but mice are not high risk, high reward seekers. They rather go with low risk, low reward. Is that a fair conclusion of this plot?
No, because it's at the time they are choosing, the risk is the same, right? Like there's this kind of tricky distinction between being like risk seeking and being information seeking. And that's always been a tension, right? Because I think it's like across species, there are differences in risk seeking. There's differences across sexes in people. There's like trait stuff in humans. I don't think anyone has yet done a comprehensive enough study to know how well like risk seeking and information seeking align. Because at the time that you choose the information, you have the same physical risk of like getting the water or not getting the water. So they're slightly different.
I thought the information here is low reward in the sense that's just one drop of water. Well, at least in your example, like in the lowest.
Oh,
case.
no, sorry. So things are still probabilistic here, right? So it's still like a 50 or 25% chance of one drop of water or nothing on the info side, and 50% chance of 4 drops of water or no drops on the no info side.
So in both of them,
the cartoon is sort of misleading.
Right, the probability of receiving reward is still the same in both.
Yeah.
It's just that in one case it's fully informed by the odor, in the other it's not.
And in this case, the amounts are different. So it actually is less water if they choose the information side.
Yeah, okay.
Yeah. Okay.
So that's why it's not the high risk, high reward interpretation that I had.
I think so. Okay, and then so we fit a decision model. If you do like a classic kind of RL Rescorla-Wagner thing, it turns out that particularly in these experiments, you can't just have an RL term that depends on water reward. We added in, to get the model to fit, a second kind of separate additive RL term for information value that suggests that maybe they're actually doing kind of an additional computation of information value in making decisions to go to the left or go to the right. And then moreover, we wonder kind of what else does this information preference depend on? And we consider that it might depend on the amount of uncertainty that information resolves, right? Like that's kind of what information value is. One way you can manipulate that is that uncertainty is sort of a state that persists over time. And so we could manipulate the delay in the task to control the duration of either knowledge or uncertainty that the animal would have. And in that case, we found, and we did a sort of similar stair-stepped, like multi-session titration of the delay in the task, and we found that the longer the delay, the more mice wanted information. So either the longer period they could be in a state of knowledge, or the longer period they could avoid uncertainty, the more they wanted to know what was going to happen. And yeah.
Sorry, I'm gonna have a lot of questions because it's something I'm really interested in. What if the information was not, so you're pointing out that it actually is not actionable. Like it doesn't change anything about like what the mouse does. But it kind of feels to me that if the information was just like, okay, a red light is going to turn on or a blue light is going to turn on, and it has nothing to do with anything, I can't really imagine mice would care about that, right? So somehow it seems like it has to be related to reward in some way, even though it's not actually telling you anything useful about that reward. Is that right?
Yeah, I mean, this is what I said earlier, like there's things I like about this task and things I don't like. One of the things that is an aspect of any paradigm like this is that it is information about a thing that the animal cares about.
Yeah.
Right? For some other reason. In this case, they're thirsty. The information is about water. In like the shock case or the gambling loss case with people, it's information about a bad thing that you care about for its own reason. And that kind of gets tied up in, well, okay, how is the information really useless about nothing, right? Like there are these sort of, there's valence to it. There's the kind of affect that it induces in the animal, right? People think the theoretical proposals are that all plays a role in addition to kind of pure reduction of uncertainty as like a motive for information seeking. What I want.
Oh, go ahead, please.
Oh yeah, no, what I want to do is get to, this was kind of an initial step, right? Of like, do mice even do this? Can we get in the circuit? But in the long term, I think if you really want to understand and ask about like, is there a desire for like predictive information that is predictive and provides structure in the world, kind of purely pure, right? Like structure of, am I going to get a red light or a blue light? Or is the flavor of the water I'm going to get that is exactly equivalently valuable to me, strawberry or lemon? I think that's a bit harder to ask, but would be a better system to ask whether it is purely about. And then you get in this whole, like, that's why we kind of lean on this evolutionary argument, right? Because if the animal is in a situation where it knows that understanding that structure gets it nothing, even in the long term, would it kind of learn not to care? Right? Like we think there's this kind of evolved heuristic that like learning the predictive structure of your world is kind of hardwired to be incentivized because in like an 80-year lifespan, that's eventually probably useful to you, right?
So I wonder if the unifying idea there might be that they want to reduce uncertainty about their own body state. Like so they want to know whether they're going to be thirsty in the future or not, and that's useful even if the answer is yes. Like you still would want to know that even though you don't like that outcome, as opposed to something irrelevant about the environment. Maybe that's why they might care about that. Maybe it would be nice to have some theoretical construct where you could kind of like figure out exactly why they do these things. I don't know if that's possible.
Yeah, I'm trying to think. I mean, there are kind of different theoretical models that are like very, very basic decision model kind of takes inspiration from. And I think some of them build in a bit of a like hedonic component. There's one theory about, well, what is the motive? Like, why do they seek information in this kind of paradigm is that you anticipate there's joy of anticipation of future good events, and getting an informative cue that kind of confirms that and locks it in is a prediction error that boosts that anticipation. And like, that's the mechanism in this task version. And I think without sufficient neural data and sufficient experiments like outside of this paradigm, it's very hard to like disprove something like that. Like that fits the data pretty well, but so do some other theories.
There's also these ideas from like music where people like music that is predictable but not too predictable. So like if it's just like really kind of repetitive, it's almost kind of annoying, right?
Yeah.
Like a kid's song or something, although kids like kids songs for that reason. But like,
Yeah.
but it also shouldn't be totally random either, right? Like that's also kind of annoying if it just like it violates all my expectations. And that's an interesting case, I think, in this light, because it actually it's not clear why that would have any relevance to me. Like hearing a song, like why should I care about that song? But
yeah, totally. It's like this other U-shaped curve. There's like the novelty U-shaped curve. There's kind of the interest U-shaped curve, the curiosity U-shaped curve. Like one of the things I want to do long term that in like a mouse odor paradigm is I think there's also something about kind of learnability and like how easy it is to fit things into your existing knowledge and structure of the world, right? Like if, right, like there's all this psych research on like, if you won't believe evidence that is going to require you to totally reconfigure your conception of the world, right? Like that just, no, absolutely unacceptable knowledge. You wouldn't want it. But anyway, yeah, we're getting way sidetracked.
Okay.
Make some progress. Yeah. Okay. Right. So there's this delay thing. Okay. So now that's kind of wraps up the neural data or the behavioral data, right? So we convinced ourselves that in this kind of task, mice want this kind of information. And we want to ask if we can have a starting point and get into the circuit and find some representation of of information as kind of its own kind of value. And so we did this longitudinal 1-photon miniature microscope imaging in the orbitofrontal cortex of the mouse. And so here, here's the video of what that looks like. This is where our lens was. Here's the GCamp virus we injected. And the reason we chose OFC as like an initial starting point is because OFC is thought to represent, among many things like task structure, the value of choice options during decision making. It also gets a strong direct projection from primary olfactory cortex, right? So there was earlier work in the lab that we know a lot about how odor identity, but not its value, gets represented in piriform cortex that we think kind of goes directly to orbitofrontal cortex, where you have little response to odors that are meaningless and then a representation of kind of associated value emerge in OFC. And so we thought that was a good place to look for responses to these odors in our task that have different kinds of value. And there was also data from our non-human primate collaborators from monkeys doing a very similar task with single unit recordings that individual OFC cells were sensitive to the prediction of information. So we thought this was a great place to look. And so we imaged simultaneously hundreds of presumptive excitatory OFC cells in 7 animals throughout the training on this task. And then we registered presumptive cells ROIs across sessions such that we think, track the activity of cells across the training in our task.
So that whole M2 part is gone, but I guess because it's unilateral, you didn't see any changes in mouses, movement, behavior, nothing?
No, they seem fine. Like the reaction time stuff is still there. They're still pretty quick. Yeah, I mean, it looks like an enormous hole, and our lenses are pretty big. They're 1 millimeter diameter lenses, but they seem okay.
I mean, it's also unilateral, so
yeah,
I suppose some compensatory mechanism perhaps happens. Yeah.
Yeah, and we implanted the same side on every animal, but then across animals we counterbalance which side is which, and then do this reversal, right?
Right.
Okay. So initially, I think the kind of most interesting thing is the responses to these center port odors, right? So these are the odors the mouse gets at the beginning. And we first looked at the ones, and part of the reason we had these forced directed trials is so that we could look at the responses to these odors, one of which predicts information, one of which predicts no information. And we look at this with sessions balanced across the reversal such that we are pooling responses. When we look at like an information prediction response, it's actually pooled across 2 different odors with the mouse going to either side, such that we can separate and be looking at not responses to odor identity or going to the left or the right, but really the prediction of information. And so here are the responses. This is kind of odor on across the 1,138 cells we recorded to the information predicting odor. There's the no information predicting odor. You can read a cell across, and then the difference between them. And I should say that we saw kind of an enormous amount of mixed selectivity. Nearly all of the, not all, but most of the cells that responded to our information predicting odors were also sensitive to whether the mouse was going to the right or the left, right? Which many people, I think, in a freely moving task like this have seen these kinds of direction or, choice movement selected activity. And so we're balancing across those side directions to account for that. And then we're looking at, and we decided to think about that the brain could encode the prediction of information with increased response to information versus no information, or increased response to no information versus information. It's just that there needs to be differential encoding balanced across the side direction. And so we found that 20% of the, and this is, we're not doing any kind of like task selection or anything. We're just saying all of the cells we can see in our miniscope, 20% of them have this differential activity between information and no information. And we quantified this by coming up with a coding index that sort of accounts for the fact that this difference could be in either direction. And we wanted it to be consistent across trials such that a cell that always that responds to information more does that consistently. So we came up with this sort of sign balanced, cross-validated coding index. And we see that this increases across the population after the center port odor. And so we kind of think about this response differentiating information from no information prediction as a representation of predicted information in OFC. But, I mentioned this thing about the mixed selectivity, and so we wanted to look at this again in a way that can kind of decompose and isolate information encoding from the left-right side encoding. And so we took that difference between information and no information activity, and we took the principal components of that. And we found like this one single PC, the first PC accounted for like 80% of the variance in the differential information predictive activity. And if we project the neural activity on every trial along that PC, we see a really nice trial by trial separation that sort of defines this axis of kind of disambiguated information encoding in the population activity. And if this really is an information prediction signal, we should be able to decode choices to get information and to seek information. And so we did a decoding analysis where we trained a linear classifier on the population activity. In this case, we're exactly balancing information left trials, information right trials with no information right, no information left trials. These black lines are decoding accuracy after the center port odor of information versus no information trials in each animal. And the purple line is then if we create a pseudo population of cells across all the different animals. And so these are those directed force trials. We're at, an average accuracy in kind of the 1 second after the odor of about 70% on these force trials. If we do the same analysis on the free choice trials, in which there are fewer trials because they very rarely choose no information, but we still precisely balance, we're at about 68% accuracy. And so we think that this, yeah, go ahead.
Sorry, this is just the classic question that, I and everybody had struggled with, but like, how do you know this is information coding and not that the mouse is literally doing something different during this period of time? So let me back up and say, what is zero? So center port order is time 0. Mm-hmm. And then if it's a force left, they're going to be moving to the left port during this period of time, right? And if it's a force right, okay. And so could that be potentially an explanation that you're actually decoding which direction the mouse is moving rather than the presence of information per se?
Yeah, so that's why you have to very carefully and exactly balance the left and right trials. So this classifier is trained on an exactly even number of left-right trials. And so it's sort of robust to that kind of confound of like going
But
to
I
the
thought-
left or going to the right.
I thought that the information port is always on the same side.
No, so in this case, all of the neural data are equal amounts for each animal of information on the left and on the right.
Because
And
they
those are from-
reverse it.
Yeah,
That's
because
from
there's the
before and
reverse.
after the reversal.
Yeah, yeah. And so that's how we've kind of dealt with, like, it's nicer if you have those all within the same session, right? But
I
because-
have a similar question.
Yeah,
But so I know you have reversed it. So that was the direction is not my concern. But my concern is that when it is informative, I can imagine the animal running faster, right? So it could be the speed. So like it's very like, so
Yeah,
how
yeah,
do you
no.
control for?
reaction times are different. And we take that both as, yes, they like information more, they go there faster, but it does create this motor concern. And so we did a control where if that were the case, you would expect, I think, on like a trial by trial basis, like a trial where the mouse is moving faster, increased neural activity or some connection between the neural activity and the actual speed, like per trial. And we didn't see that. So I think that's the kind of strongest thing and also the kind of emergence of like when the differential coding happens aligns to receipt of the odor rather than when they start to move to go to the port or when they get to the port. It seems to be kind of time locked to, I know it's not precisely time locked to like when the odor comes. I think that's and we did kind of actually measure like when does it get to the mouse's nose. I think that's a reflection. That's, I think, true about odor responses in OFC generally with such a short, this is only a 200 millisecond odor pulse.
How
But
do you
the-
measure their movement? Do you have some video recording?
We have video recording, but we also like each of the ports has an IR beam break. So we don't have like vigor during their, I mean, I do have video tracking, but we have the time they exit the center and the time they enter the side.
So you don't have the exact movement kinematics here, like how they run, how fast, like what they like. It's not controlled for that. It's more controlled for the reaction time. Like when did I get to the side port?
Yeah, they're pretty quick though. So the reaction times are on the order of like 300 milliseconds. They're pretty much going straight from the center to the side.
I mean, I'm a head fix person, so for me, like
Yeah.
thinking about all the uncontrolled variables, that drives me crazy. But then, yeah, I also don't know how much OFC is encoding. How much OFC encodes like movement kinematics? Like, do we know about those?
I mean, there is this thing and this kind of the like choice direction and like where like this side thing comes up a lot in freely moving rodent tasks in OFC. It doesn't come up as much in the primate work, right? Like when people have done the kind of classic like primate decision OFC stuff, because within a session, the target the animal saccades to is in a different spot on every trial. It doesn't matter as much, and I think people don't look as much, but when they do, they don't see as strong a direction signal. But I don't know if anyone's done a careful, like, is it these paws and this body movement versus this one?
Yeah.
I should say, I think our decoder is kind of the best evidence of that because, it's predicting with really high accuracy. Is it an information or no information trial? It can do that equally well across the two sides. And there are trials where like the reaction times are similar, kind of on the far end, and it can still do those. Like it's not, I think, categorically failing on like the faster no information trials.
So I just want to add a note that I know we started late with all the technical things, but let's aim to finish in like 15 minutes.
Yes, I think
Thank
we
you.
should be able to do that. Okay.
And it's completely our fault for asking so many questions, Jen.
No,
That's
no,
only
it's
'cause
good.
we're
I love
interested.
it. I'd rather have a discussion than me like droning on. Okay, so our decoder, we're using the decoder to kind of argue that this is a signal that lets you see the animal's choice to get information on a trial by trial basis, as if it's really about this like choice to go and get information. But we wanted to know, and there's kind of a classical thing as one talks about signals of value in decision making, is it a signal of information prediction, or does it scale with the kind of cognitive intrinsic value of information? And you can look at value of information in different ways. We did this experiment that was kind of possible to do for the mice, where we changed the delay in the task. And so here is the information coding index across the cells in the version, our original long 10-second delay version. And then when we look in sessions with the 1-second delay, when mice became indifferent to information, the magnitude of this coding difference between information prediction and no information prediction decreased, which suggests to us that this is truly a signal of predicted information value, not just information, no information, but it really does matter how much the animal wants it and how much information there is. And we did some controls that, this, because we're doing this across sessions, this doesn't seem to be just about time elapsing or our alignment across time, or the fact that the entire task is shorter. It really does seem to be a scaling of this response to the odors at the center port that predict information. Okay, and so there's lots of different value signals in this task. I've talked a lot about these center port odors that predict information. And we've made this kind of analogy that, they are predictors of information. They're kind of conditioned stimuli for information. And then the odors that are giving information at the side port are, by analogy, an unconditioned stimulus of information. And we also have odors that, like we talked about before, predict water or no water, are kind of classical, more conventional conditioned stimuli for water reward, and then the water reward itself. And we saw neural responses that are kind of consistent with these different types of value representations of all of these. Those data are, in the supplement, they're in the paper. But one thing that's really interesting that I wanted to touch on briefly is these odors on the information side port that are both water predictors and providers of information. And we see neural responses that reflect both of those aspects in individual cells and across the population. So this is an example cell that responds in a kind of consistent way to both the odor that predicts water and the odor that predicts no water at all. And then we have other cells that respond in, I think, a kind of more conventional way, most to the odor that predicts water, intermediate to the two no information odors, and least to the odor that predicts no water. Yeah.
Question. So I really liked that you broke it down to like odor providing, like there are really multiple things and multiple predictions going on here. The initial plot that you showed us, the key plot, I guess, that OFC can basically decode information versus non-information prediction. That was done during the center odor, but the odor that is free choice, and then you decode on trials. So that was the free choice odor. I just want to make sure that I'm
We
getting
did
that
both.
right.
The one I showed you happens to be the forced
Yeah.
no info, but it looks pretty reasonably similar on the free choice ones.
But the forced info, one might say that's a sensory decoding. That well, no, you do reverse the odors, right? So all of that is controlled, right? But then if you do because the free choice is perhaps the cleaner version, because there is like it's the same sensory. Then you still see that you can.
Yep.
Well, but the very you can still decode information versus non-information. We
Yeah,
love.
so here's the free choice. So,
I think we're seeing your calendar.
Oh no, oh no.
It's good you're camping.
I know. Up to the Adirondacks. Okay, yeah.
It's okay, not the whole
here's kind of the
YouTube season.
average accuracy in like this 1 second on the forced versus choice. Here's, on a per mouse basis, it gets slightly worse for the free choice. One could say either that's because the signal is weaker, there's way fewer trials. Like we even had to throw out a mouse because it chose no info so infrequently that you can't train the classifier. Yeah, but it's true. And actually I'm gonna make a lot of hay out of the fact that we have like the choice information odor and then the no information predictive odor and the forced information odor and the no information predictive odor. So you can train decoders on all of those things.
Okay, thanks. We are not seeing again back to your calendar.
Yes, I'm going. Yeah, no, I'm just
Okay.
going back to where I was in the slides. We're almost there. We're making such good progress. Okay, we did that. We did these side port odors.
We are seeing your calendar.
Yes, I'm back now.
And
Right?
then
Now
we
you
are
see my
not
notes.
Yeah, exactly. Awesome.
Okay.
Thanks.
Okay, right. So there's the side odor responses were really cool because we were totally surprised. Like we've done in the lab, here's a mouse, train it, odor A predicts water, odor B predicts no water. You get basically no response in OFC to odor B because it is a CS minus for water. The mouse doesn't like it. It doesn't care. It has no value. In our task, we had this fairly widespread, like 25% of the cells, I think, have this response where there's a correlated response to the two odors that give information, even though one of them means no water and the mice hate it. And that was surprising. We thought that was really cool and interesting because it says that the brain finds something in common to these two opposite valence signals because they're both giving the mouse information. And there's a contrast of that with the no information odors. And I'll come back to that at the very, very end if we have time. But okay, last big question is this its own thing, right? Like at the level of the dopamine neurons, if you look at a person doing like a curiosity task in an MRI scanner, the brain, the kind of common reward system lights up. And in the monkey, it's even the exact same neuron is responding to juice and information. Is this signal in OFC? And I think this was more true several years ago, but there was sort of a debate of, well, in these value-based decision-making paradigms, is OFC indicating kind of common currency, like complete integrated subjective value of an option, or is it signaling and having multiple representations of like different aspects of a choice's value? And so we wanted to answer that, and we looked within the original task, but I wanted to talk to you guys about an additional task we did to have odors that predict information value and odors that predict physiological water reward value in the same epoch in the task so that we could directly compare them. And so we took mice that had trained in our task that were seeking information and had learned there's an odor that predicts information, there's an odor that predicts no information. And then we added in 2 additional trial types with additional odors. So we added an odor that predicts a certain large value, large amount of water reward, and an odor that predicts a small value water reward. And then we looked at responses, this differential encoding of information prediction and differential encoding of water value prediction. And we also did a reversal in this task, so we're controlling for side direction, for odor identity, etc. Right? And the first thing is that if we just look at, oh, this is going to get complicated really fast. Okay, so look at the plot on the right. If we ask, okay, is there a correlation across the population in the cells encoding of an information prediction response and a water value prediction response? There's a somewhat small, modest correlation. Then we went back and we looked at, and I should show you that the responses, oh, if I do this, I'm gonna give you back my calendar for like 1 second. Hang on. responses to the forced and choice information odors. So this is the information forced odor, and this is the free choice trials when the mouse chooses information. That neural activity is incredibly similar. It's correlated like 90%. They're very, very similar. We made this analogy with this task where there is odor predicting water value and odor predicting information value, that if everything is one kind of generalized value, the neural encoding of this value in response to these odors should be similar to neural encoding of information value in response to these odors. In fact, perhaps as similar as encoding of information prediction in response to the forced and choice odors. Does that logic make sense? It's a little convoluted. And so then we looked at, okay, well, what about the responses to the information forced and information choice CSs? And those are really highly correlated. They're 3 times more correlated than information prediction versus water prediction. And we did kind of a shuffling, like a bootstrapping analysis. And it's very, very unlikely for this level of difference to emerge by chance, which suggests that information prediction is kind of its own thing. But to get at that further, we went back to decoding, and we again trained this information prediction decoder, right? That's the purple line. We're able to do that. Then we trained a second totally separate decoder on side-balanced high-value water trials versus low-value water trials. We're also able to decode water prediction. These are, again, all at kind of the same point in the task. And then what we asked is, let me come over here, are the cell's contributions to decoding water and decoding information related? And they're not. They're uncorrelated, right? So the cells in the neural population that are important for encoding predicted water value are pretty different from those that contribute to encoding predicted information value. And if you look at, so we're training decoders, they each have kind of a hyperplane across the high dimensional population. And the kind of logic is that if things are encoded really similarly for a downstream region to read out, you might expect those hyperplanes to be sort of parallel and aligned. And instead, here they're nearly orthogonal. They're at like 99 degrees apart. And that suggests, again, that something like a downstream area reading this out, or at the level of the neural population, they're separable, distinct representations of the information prediction and the water value prediction. And that orthogonality is really different from what we see in comparison to this like forced choice distinction. Okay. And I want to be mindful of time. I told you that mice value information. We see a representation of predicted information that we can decode in orbitofrontal cortex. The magnitude of that signal scales with the uncertainty-based value of that information, and it is distinctly decodable from the representation of predicted water value. And I didn't get to the learning stuff, but this idea of there being a CS and a US for information, we think we can kind of see those signals emerge during learning, and we think there's reinforcement of the information predicting odor, and our kind of future goals are to figure out how and where that's happening and really work through the mechanisms in the circuit.
Could you, we're not full screen.
Oh my goodness, I've been showing you my notes.
Well, most of it was visible, but I wanted to make sure we have your summary slide.
Yeah, totally. Sorry.
So the first one, we saw it. The second point also, we saw it. The third one, predicted information scales with value of information. Which one was this?
That's the delay thing, right? We didn't do kind of a full titration. We just looked at like, okay, well, sessions where they aren't wanting information because there's a short delay, and then the longer delay sessions.
Because the longer delay is basically more uncertainty. So it's like, so that's why now perhaps the certain option becomes more wanted, right? Because if you add long delay, plus uncertain, plus the lack of information, no information one, then it's too much uncertainty. So that's how I saw it. So,
Yeah, I think that's fair. Someone had a question.
Yeah, hey,
Hi.
great talk. Thank you.
Thank you.
I have a question about the OFC neurons. So, as far as I understood, you did calcium imaging there.
Mhm.
So this is not dopamine specific. It's like you basically imaged all cells.
Yeah, and in
Okay.
fact, we think we're not looking at like dopamine or prediction areas. We're just looking at presumptive mostly excitatory OFC neurons.
Okay. And then you mentioned in your supplementary that I didn't read that there is both responses in some cells to the cue, the odor, but also I was interested, is there also responses to the reward itself? You haven't really talked about that. So can some of these neurons respond to both, like we know from the typical dopamine neurons, depending on which area that you look in?
Yeah. So, okay, let me think. We looked at, so neurons responding to the water reward itself. The wrong. Okay. Sorry, I gotta do my weird calendar thing. We'll get there. So there are cells that respond to the receipt of water reward itself, right? It's not so many, but there are some and they're pretty consistent. We looked a lot because we checked kind of extensively, and it is kind of a thing in OFC that the same cells often don't respond to like a CS and a US, like the predictor and the actual thing. So we compared directly the cells that were responding to, do I have that? I don't know where it is. The kind of water CS and information CS cells, right? The cells responding to the odors that predict water and the odors, I'm on my thing again. The odors that predict water and the odors that predict information. And those are for the most part different cells.
Mm-hmm.
Did that
Okay.
answer your question?
I think so. I'm interested in this because we're measuring from dopaminergic neurons fiber photometry, and I was just curious if in OFC there's similar dynamics like we see, for example, in the striatum, where when an animal learns, there is these cue-related responses that emerge that then predict the reward. And it's a simple paradigm, right? That dates back a long time. So I was just wondering if these signals are similar in OFC. And of course, these animals are highly overtrained, so it's hard to say.
Yeah, I mean, we looked at the emergence during learning directly. We weren't sort of expecting like a prediction error signal so much as we looked at representations or responses to the odors that predict things and responses to the odor or the water that provide things, right?
Mm-hmm.
And we see both of those. They're not so much the same cells. They do pop up. So like the response to both of the information giving odors, the one that predicts water, the one that predicts no water, that like pops up when we introduce those odors and we associate them with water, right? I didn't look at like the trial by trial time course of that carefully. And we did sort of a lot of looking of like, are these truly like separate populations that are defined in some way? And we don't have good evidence for that, right? Everything seems to be interleaved, mixed selective, right? But you do end up in any given like task session with these kind of orthogonal population encodings.
Okay, thank you.
Thank you.
Jen, do you think you would have seen the same things if you recorded somewhere else besides OFC?
That's a good question, right? Like value is everywhere. I'm not sure what you're getting at. I mean, the thing that we're really interested in is, can we find somewhere that is recognizing that a thing is information, like regardless of its valence, and conveying that that's a good thing. And so I'm kind of hunting around for that signal now, recording other places. I don't have the data yet, though, right? Like for sure in piriform cortex, we wouldn't have seen the same thing, right?
Maybe.
And
How do we
the yeah, I should do it before I say that.
You're saying for sure you would not have seen it.
Yeah, exactly.
Why are you so sure?
Well, if you, in our lab's hands, if you do a more simple, like water conditioning paradigm where there's like 2 odors that predict water and 2 that predict no water, the signals are really different. That was that signal I talked about before where there's sort of, you get responses to the water CS plus odors that kind of disregard identity and no response to the CS minus odors. And that's different from what we see in this task. Oh, sorry, that's in OFC. In piriform cortex, you get just like the odor identity.
Yeah.
What 2% of cells.
Just a quick question, methodological. Why do you guys do necessarily miniscope? So why not recording so you don't have to also remove a big chunk of the cortex?
Like
it doesn't seem like you're doing like cell specificity or projection path, so it's just general recording. So is there a reason you prefer miniscope?
To like a neuropixel?
Yeah.
I mean, when I started doing this, people didn't do chronic neuropixels or freely moving mice, right? So we started freely moving, and the original pitch, at least for me coming from flies, was, well, if we're imaging, we're going to be able to watch the same cells and ask, you know, how does this cell change? Is the same cell responding to information?
But I don't think you showed any data on cell matching across days, or did I miss something?
I didn't show it. We did it. And
Right.
so like all of the data I showed you were pooled across days, the kind of validation that, you know, the cells are the same and that there's consistency in their encoding across time is all like in the supplement.
Right. How many mice went into this study?
The main imaging data are 7 animals, and then there's another 6 in like the 4 odor value task. I think everything else is kind of pooled from those guys.
Because you had to do the miniscope reversal one side, the other, then
Yeah.
Cool. All right. Thanks a lot, Jennifer.
Thank you guys so much.
Really,
I really appreciate the interest.
Yeah, it was very insightful and inspirational. I loved it. The topic of information seeking is a philosophical topic, so there's a lot to discuss there. That was awesome. Thank you.
Thank you guys so much. Reach out if you have further questions. Happy to talk more. Thanks.
Have a great day.